Good afternoon, ladies and gentlemen. On behalf of the Hearing Health Foundation, I'm honored to welcome you all today for a truly important event. I'm Dr. George Gates, the Scientific Director of the Hearing Restoration Project, and today we will be discussing the promise of cell regeneration and how a group of scientists hoped to unlock a cure for hearing loss in the next 10 years. I'm proud to tell you the good news. We have assembled a dream team of scientists and researchers under the Hearing Restoration Project with one goal, and that is to develop a biologic cure for sensorineural hearing loss. Today you'll hear from several in the consortium who will tell you why we are so optimistic about this outcome. Collaboration is clearly the key to enhancing progress. It's my greatest hope that the HRP, as we call this, will be the way to find a cure, which, as we all know, will be a huge relief to the nearly 40 million American people who already suffer this unfathomable loss. It is thanks to the generosity of the Hearing Health Foundation and the Board of Directors that we are gathered here today. Our goal for the next decade is to raise $50 million in order to pursue this cutting-edge scientific research. We need public support to get there and need to make hearing research a national priority. We are delighted to have Dr. Michael LaPook of CBS News with us today. He is one of the most recognizable doctors in the country, filing regular reports on the CBS Evening News. He most recently filed a report on the rise of hearing loss among adolescents, and now he will lead a panel discussion on cell regeneration with some of the leading doctors and scientists in the field. We all know it will be an educational and inspirational event for us all. Dr. LaPook.
Let's see if does this pick me up? All right? Okay. So I'm John LaPook. I'm the medical correspondent for the CBS Evening News with Scott Pelley. And I'm an internist, and I'm also a gastroenterologist. So that you might think that makes me a dilettante with all those different jobs. In the morning, I see patients for internal medicine and GI, and in the afternoon, I go over to CBS. And I'm so glad and honored to be invited here because as a mea culpa, I will tell you that one story, which was about hearing loss in our kids, that's caused by the fact that they have various digital devices that are blasting their brains off, was the one and only article we've done on hearing loss in, I would say, over 350 that I've done in the more than 5 years since I've been the medical correspondent at CBS. So clearly this is a huge problem. You'll hear about this more. But why is that? Why is that there's not more coverage for this problem? I took this as an opportunity to do the deep dive, as I like to do, and read a bunch of background articles about this, and I'm thankful to Dr. Mark Parker, who helped bring me along and wrote some review articles and spoke to me on the phone to give me my Reader's Digest version so that I would understand what the latest and greatest is in the field. I think with that as a brief introduction, I'm going to go right to our panelists. The first one is Dr. Sujana Chandrasekar, who is director of the New York Otology, and he's also the chair of the Board of Governors of the American Academy of Otolaryngology, Head and Neck Surgery. And also, she and I have shared a patient recently, and I can tell you that clinically she's a wonderful doctor. And the last thing I will just say, in my doing the deep dive and my doing diligence about today, I checked out the panel here because I'm not an expert in this field, and I can tell you that the people I spoke to, this is the creme de la creme. These are really, this is the A-team, and people were very excited about who was on this list. So maybe you all know that already. I'm still a neophyte and learning, so you should be very excited about who you're about to hear from. They are really the leaders in the field.
Thank you. I am representing the clinical ENTs on the panel. So I do clinical otology and neurotology on the Upper East Side of Manhattan. I'm at the VA hospital in the Bronx once a week. My interests are in treating current hearing and balance disorders and doing what I can to help prevent and treat differently in the future. I owe a lot of what I know to Dr. Noel Cohen, who's in the audience. He was my chairman at NYU when I was a resident, and his grading of my papers usually were like, where'd you come up with this idea? But even so, I've gotten where I've gotten, and I'm very happy to be here.
All right, and what we're going to do is I'm going to introduce briefly the panelists. They'll tell you a little bit about themselves and their work, and then I'm going to do some Q&A where I'll ask them more specific questions about the specific research that they're doing. So thank you, Sujana. Our next panelist is Dr. Ed Rubel, who is from the University of Washington, and by all accounts, the person who's really leading this HRP effort. He, along with Doug Kotanch, was a person who made a seminal discovery in this field, which was that in chicks, in birds, when their hair fibers died, they grew new ones. And nobody knew that before that. We don't know still why, and it's going to be a question I'm going to pose to him is, what about us mammals? What about it? What is it in evolution that made it that we could survive without those hair cells? Maybe we have other ways of surviving, but in birds they needed those and they died and didn't reproduce if they lost those crucial cells. But it's a fascinating and completely unexpected finding that he made and has since gone on to further research. So on a personal note, I will say when I saw his name listed, I said, how many Ed Rubels can there be? And I looked and clicked on his picture in Google, and I said, yeah, same Ed Rubel who taught me when I was a senior at Yale in the fall of 1974. And I rummaged through my papers and found this paper, an annotated bibliography, Effects of Testosterone on Sexual Differentiation of the Brain, compiled by Jonathan LaPook, no MD back then, for Psychology 90-7A, Ed Rubel, December 18th, 1974. And I sprung this on him beforehand, and I wanted to set a Guinness Book of World Records for the most amount of time, which would be, 37 years, between the paper being handed in and the student grade grubbing, because there's no grade on this, and I'm not saying anything, but all right, anyway, I'll leave it to your discretion.
We'll see how well you handle the panel.
Okay. Anyway, I will say, and then I'll turn it over to him, that seriously, because when does that ever happen, right? Back then, he had just gotten his Ph.D. about, so 4 years earlier, and I had been taking all these pre-med courses in cell biology, and I had the latest and greatest from the Yale cell biology people and the biochemists, and it was all in my head. He was brimming with excitement over the fact that there was stuff inside the cell that might influence neural development and neural processes, and I could see the silhouette then of what, I didn't predict this type of greatness in terms of a career, but he was so excited, and it was always coming up to me after class, and we would sit and talk, and he would want to know, what did Joel Rosenbaum say about the way this works and about that works? So I saw that excitement then, and it was so exciting for me to then see that, oh my gosh, that makes perfect sense. He went on to this distinguished career, and he went out and learned more about cell biology than I'll ever know in a lifetime, and now he's sitting here about to tell you about his work. So Ed Rubel.
Oh, thank you. I guess I can call you John rather than Dr. LaPook. It's a pleasure to be here, and thank you, and thank you. This is a wonderful moment for me. As John said, many years ago we discovered hair cell regeneration in birds, and I've continued to work in that field. I'm a professor at the University of Washington in Seattle, about as far away as we can get, and but not elect in terms of our interests. I'm in the departments of otolaryngology, head and neck surgery, and physiology and biophysics and psychology. And my work is primarily on the development of plasticity of the peripheral and the central auditory system. And the work we'll talk about today, of course, is on regeneration, but we're also interested in why the cells die, and can we figure out ways to make it so they don't die and so we don't lose our hearing. But we'll talk about regeneration, and I'm delighted to be here.
Thanks. Dr. Stefan Heller, I need my notes for this because I know him less well than I know Ed in terms of a background, from Stanford University School of Medicine, is professor in the Department of Otolaryngology, head of neck surgery and molecular and cellular physiology at Stanford, and an associate member, Gesundheit, of the Stanford Institute for Stem Cell Biology and Regenerative Medicine. His expertise is in stem cells, human embryonic stem cells. He's diving into them, seeing how they can be teased forward, perhaps into some sort of useful cell that can be used in the treatment of hearing loss. I'll turn it over to him.
Hello, thank you, John. I have a history to New York. I was a postdoc in New York starting 16 years ago. For 5 years I was working at Rockefeller University. Then I got my own lab at Mass Sinai Infirmary up in Boston for 5 years, where I became interested in making hair cells from scratch, trying to figure out how to make hair cells or inner ear cell types in a culture dish. So I tried to learn how to do that or teach myself how to do that. And over time, I think we have developed the cell biology that lets us control inner ear development in a culture dish. And I'm going to talk a little bit more later about that. And I'm looking forward to the whole afternoon. Thank you.
So stem cells is a very stem cell, that's a very sexy term. We all have heard it in the media. Developmental bio, not so much. But to developmental biologists and to the people working in this field and rolling up their sleeves, really you need to know, in order to use stem cells, in order to be able to do anything that's clinically appropriate, clinically relevant, you got to know how the cell works. And so Dr. Andy Groves is from the Baylor College of Medicine. He's an associate professor there, and he uses the inner ear as a model system to address fundamental questions in developmental biology and regeneration. How do these cells start off, and how do they get from here to there? And if there is someplace we don't want it to be, how can we nudge it a little bit and get it to go over here? So, Andy, take it away.
Thanks, John. So yes, as you heard, I am from Baylor College of Medicine in Houston, Texas. As you can hear, I'm not a native of Texas, but I very much enjoy working there. My background is in developmental biology, so I am interested in how different tissues and organs form during embryonic development, and my lab focuses particularly on the development of the inner ear. And the reason I'm here today is that I believe that by understanding how a tissue, in this case the inner ear, is built, it may offer us some clues as to how that same tissue may be repaired in the adult after damage that obviously results in hearing loss. So I bring the experience of a developmental biologist and an embryologist to this problem. And it is in fact Ed Rubel who helped get me into the regeneration field about 15 years ago. So I'm sort of sitting here as his scientific son. And I'm hopefully not a prodigal son. So I'm very happy to be here too.
All right. So I think you'll see that it's actually a lot of thought went into this panel. Not only are they leaders in the field, but they're from different areas so that it's going to all come together, hopefully, to give you a picture of where the field has been, where it's going, what's hype, what's hope, what's real, what's imagined. And so with that, I'm going to start it off, I think, with an overview. Many of you probably know this already, but I'm going to turn to Sujana to give us a clinical overview. I think people don't understand how big of a problem this is in America. And we were talking beforehand, in fact, there is a stigma attached to hearing loss. People, it's kind of okay, we're saying that it's okay to make fun of somebody kind of in media and out in the world of somebody with hearing loss. You would never make fun of somebody who's blindness. So what is it about that? How does that affect us in the real life? I'm an internist. I see patients who are elderly. And one of the most common things that has happened in terms of an early symptom of hearing loss is not so much that the person will come and say, I can't hear, although they'll do that too. I'm in a room and I can't quite make out what people are saying, especially if there's a lot of noise. They can't discriminate. But surprisingly to me earlier in my career, and not so much now because I'm used to it, the family will come and say, you know, Grandma, we think she's a little bit demented. We think she's losing it. And it's not that she's demented, it's that she's hard of hearing, so she's not picking up the nuance of the conversation. There is a stigma attached to it, so she's not coming forward saying, I'm deaf, or I'm having trouble hearing, I need help. So she tries to fake it, and people aren't particularly good at faking it. Conversation is very nuanced, and so what the person on the other end of that conversation hears is somebody who's not quite picking it up, and they think, oh, maybe they're just losing it. So this is a really big problem. We talk about Alzheimer's, 5.3 million people now. By the year 2050, they're going to be 15 million. Why? Because the population is aging. Well, guess what? This is a problem. Hearing disorder is a problem of older age, and it's just going to get more and more prevalent. So with that, I will turn it over to Sujana.
So I think some of the easiest numbers to remember are there are about 36 or 37 million people in the United States who have enough hearing loss that it affects their life. So they are either not really following conversations or picking up enough to sort of fake it for a while, or in fact, they end up withdrawing from social activities. Of those 37 million people, only 20% actually use hearing devices or hearing aids that we have now, which means that 80% of people who could benefit from the current technology are not taking advantage of it or not able to take advantage of it for many reasons. Some of it is the social stigma. As Dr. LaPook said, it's sort of made fun of in the media, it's made fun of in jokes. There's a social ability to make fun of somebody because they can't hear, where you really wouldn't make fun of somebody because they couldn't see. There's the stigma of, oh, if I get a hearing aid, everybody will know that I'm old. And I will say, but now everybody knows that you're deaf. So, you know, you might as well get a hearing aid and be able to participate in the conversation. A lot of people come in because their spouse feels that they're not hearing, or their children feel like they're kind of losing it. They feel unable to connect across generations, so that grandma or grandpa has a lot of difficulty interacting directly with the grandchildren. I've had patients who've withdrawn from very pleasurable social activities like bridge, where you're holding the cards in front of your face, so it's very difficult to lip read around the table. And they've been unable to participate in a very social, engaging game like that, and they withdraw from that. So this is a significant problem. We anticipate that in the next 20 years, that number of 36 or 37 million in the US is going to double. So we're going to be looking at 70 million people with significant hearing impairment. That's due to aging, that's due to some toxicities that we may talk about later. It's due to noise damage. If you have children in your family who are listening to their personal listening devices, sometimes you just want to yank that earbud out of their ear because you can hear the music quite loud and that must be blasting their ears. So there's a lot of causes for it, and what we need to do is identify how to prevent it, identify how to treat it, and identify how to make the treatment more beneficial to socialization.
What would you say is the biggest misconception, or one of the biggest misconceptions that you run into when somebody comes in?
So one of the hugest misconceptions is, well, of course I can't hear, I'm getting old, so therefore I'm not supposed to be able to hear. Well, that's actually not very true. We are all getting old. We just got older now than we were 5 hours ago. But, so, you know, we're all getting old. But the pleasure in life is really from hearing and actually from overhearing. And I'll sometimes talk to my patients about getting the gossip or getting the aside in a conversation, which they're missing if they're missing some of those high frequencies where the terminal consonants, the meanings of words, are located. So that misconception is, well, of course I can't hear, I'm old, is very much something that we have to fight against as physicians and as members of society.
And, you know, these days, I think there are more and more misconceptions and lack of appreciation of the nuance of language. I mean, we see it in auditory communication, but we certainly see it in emails, right? You know, if somebody writes, "That's great," that could have 8 different meanings, you know, from "that's great" to "that's great." You know, and I think that nuance, it's such a great point about the nuance of language, that it's subtle. And it can mean the difference between that and that. So, that's a great point. Ed, I think people probably, I'd love to hear just personally what it was like to be at that moment when you realized that something that everybody figured could never be true turned out to be true. So bring us back to, you're discovering that one of the things that we know is true, which is that when a hair cell dies, not the hair cell on the head, but the hair cell on the ear that's so important for hearing, when that dies, it can actually regenerate in probably everybody but mammals, right? So what was that like, just as somebody who was there?
Well, it's actually a pretty funny story. It's, so this would be 1985. I was in the otolaryngology department at Virginia, and I had a colleague named Paul Lambert, who's now at chairman of the Department of Otolaryngology at University of Medical College, South Carolina, and we had a young resident named Raul Cruz, and Raul came to work in the lab, and we decided that Raul should figure out what is the first time that we see damage due to what we know are damaging drugs. What are called gram-negative aminoglycosides or an antibiotic. We know those are damaging.
Like gentamicin.
Yeah, those are damaging to the ears of humans and all other animals. So we wanted to study that, but to understand why they're damaging, we figured we had to see the first time we could actually see damage. So we gave some chickens, and we were using the chickens because it has an easier inner ear to study. I don't need to go into the nuances of it, but it's a little easier. But it responds to these drugs in the same way as a mammal. And so we had the animals, we gave them the aminoglycosides, and we sacrificed the animals at various times after. And when we sacrificed them 8 days after, we got a lot of loss of hair cells. And Raul did the counts, the actual counts of the number of hair cells, and he showed me the data. And by 22 days, there wasn't as much loss. And I said, Raul, you must have mixed up your animals. Go back and do it again. Because he was just a resident, he didn't know what he was doing. And he brought me similar data. And then I said, well, change your counting criteria. Now, this is a technical thing. And he went and changed his counting criteria and brought me the data. And I said, well, maybe I better look in the microscope. And I went and looked in the microscope, and sure enough, the hair cells were coming back. And I thought, wow, what's this due to? What's going on here? And we just were excited, and actually during that period, then I moved to a different state, and we took a while to do it. And Doug Kotanch then at Penn, who saw the same thing after noise damage. And we reported ours, and then he reported his a few months later, and then we both published that, and then we got together for a couple of major papers showing that this was due to new cell division and the creation of new cells in the inner ear, and wow, we had a new field.
So Ed, I'm gonna ask you one more question, move along, and then come back. But I've got to ask you. So that was a quarter century ago, right?
Yeah.
So certainly by now you've sprayed a little Rogaine, a little Propecia in the ear. You've made those hair cells grow. You have this amazing discovery. Oh, that we can make them grow again. How did that change your thinking about how you would even approach it, from maybe more electrical type of things to biochemical cell biology type of solutions?
Well, we and other people recognized very early that the new hair cells were being generated by the support cells surrounding the dying hair cells, and we knew it was basically a cell biological and molecular biological problem, and that what we had to do, we actually went out and looked, maybe it's happening a little bit in mammals, but it wasn't, and we realized that what we had to do was really understand this and document and understand the changes that were going on. We also realized it was going to be a tough problem. Right. And that what we were going to have to do was bring a lot of tools to its disposal. And at that time, we didn't have anywhere near the tools that we have today in biology.
So what a perfect segue, right? Yeah. Notice how I'm doing this. To the tool guys, okay? So Dr. Heller, talk to us. We're very impatient, right? We want solutions like this, and we've got stem cells, and we should be able to walk tomorrow if you're paralyzed, and we should be able to hear tomorrow if you can't hear. You just throw in those stem cells. So give us a sense of, we've been around for millions of years. This is a microsecond on the blip of the evolutionary x-axis. People have wrong expectations about that, but I know that you in the field are exploding with excitement over where this is leading, even if it's not going as fast as other people want it to. So can you give us a sense of now, we understand this great discovery, we understand the clinical importance. Roll up your sleeves and tell us about the tool set.
Right. So nature has solved the problem already. Chickens and birds regenerate their hair cells. If you look at the whole animal kingdom, if you go to any animal museum or so, a museum of science, you see the whole pedigree of animals. 99% of animals are able to regenerate their hair cells. And they don't even go deaf at all. They lose hearing and it comes back. So nature has figured out how to do it. And for some reason, mammals like us have lost it. And we are really trying to figure out what these differences are, and Ed's findings have laid the groundwork for that. And over time, it was very clear that a quarter of a century ago, we were not there with all the tools that we needed. Molecular biology was just starting out at this time. And being able to manipulate the genome hasn't really happened in that extent as we are able to do it now. So I think we are really at the brink of a new decade where we will be using novel tools. And then one of the tools that we are using is stem cells. And this came out of a desperation in a way. I started out to learn how hair cells work, how the inner ear works. And the study of the inner ear has been hampered over time very much by this one example. If you look at a retina, the light-sensitive cells of the eye, let's say a human retina or a mouse retina, a single retina contains 140 photoreceptor cells. So a cell biologist has 140 million, sorry, I forgot that word, million, 140 million photoreceptor cells. So a cell biologist working on a retina gets a retina and has an abundance of cells to work with and to study them, and also the cells that surround these photoreceptor cells. In the ear, the mechanosensitive hair cells, the cells that you hear with, in the cochlea, it's only 15,000. 15,000 versus 140 million. Just a simple comparison, this is one of the reasons why we don't know so much about the ear, because it's more or less impossible to study it with all the biochemical tools and the molecular biology tools of the 80s and the 90s. But now we have reached a decade where the tools become more and more sensitive, where we can focus, where we can do experiments with a couple of hundred cells. And so now this discrepancy, being able to do experiments with a couple of hundred cells and not needing millions and multi-million cells has shrunken. So we are now able to do more research.
And by the way, these hair cells, you think of them like standing up like a field of wheat, And when you blast it with too much sound, they just get flattened. Literally looks like a flattened field of wheat. Right. And they die. And that's it. It doesn't come back. Sometimes it does.
No crop circles in those.
Yeah, people aren't being paid not to grow it. Not to make you cell biologists, there are stem cells and there are stem cells, and just sort of like, again, Reader's Digest, there's human embryonic stem cells. Then it turns out that there are stem cells that hang out in the adult tissues in various places throughout the body. There's also the ability of taking skin cells, putting them into a petri dish, throwing some magic over it, some little chemicals and some other things into it, and bringing them back into human embryonic stem cells, and then from there you can tease them up into any cell in the body, pretty much any cell in the body. And then recently they've been shown that you actually don't even have to do that. You can go from one adult stem cell from one type of tissue to another adult stem cell from another type of tissue, okay? What are you dealing with?
Us. We're dealing with three types of stem cells. One are embryonic stem cells, and we are working on mouse and on human embryonic stem cells. We work on mostly on the cells that you just described, the cells that you can take a skin biopsy of a mouse or of a human patient, and then I can tell you we have gotten the first skin biopsies of human patients in our lab just a month ago, and we turn these skin cells into stem cells again. And then we move forward using developmental tools like the research that my colleague Andy Groves is doing and applying this to the cells, and then we make basically an inner ear in a culture dish, in a test tube. And then we can study the ear and we can study regeneration. We're also interested in somatic stem cells. These are the stem cells that you can find all over your body. Your skin is constantly renewing. You get, there are stem cells in your skin. And the skin is not exhausting. I mean, it uses elasticity over time, but it's constantly making new cells. The inner ear cannot do that, but the inner ear of chickens can do that. In the vestibular system of the chicken, there's a turnover. Hair cells are being removed and new hair cells are being added like it is in the skin. So they are cells with stem cell characteristics, and we find some of these characteristics still in the mammalian inner ear. Even in the human inner ear, we can see some of these characteristics. They're well hidden, and then we just joked about this. It seems like these cells evolutionary got a new job, and they need to be retrained now, and that's our task in the next couple of years to redo this original job again.
So for the lay public listening to that, because I know it can sometimes get, wow, what is that? We think about stem cells because part of it is our fault in the media. You're going to cure disease with it. You're going to throw somebody's paralyzed, throw a little stem cells, they walk. They're blind, throw a little stem cells, and they can see. To scientists, to basic scientists, the really exciting thing is to be able to study disease, to have a disease in a dish. So whether it's studying hair cells or studying, for people who are studying ALS, imagine being able to take those stem cells from the skin of somebody who has ALS, and now you de-differentiate it, you tease it back down to a human embryonic stem, to an embryonic stem cell, tease it back up into a nerve cell, the kind of nerve cell that normally you would only see after the person died, and by the way, it wouldn't be alive. And now you're studying in a petri dish the disease. Live nerve cells in a dish. You can try drugs on it, you could try things that you would never be able to do in a human being. And these models are enormously exciting to basic scientists. So I just want to point that out. Stefan, Andy Groves, Dr. Andy Groves, he has the type of field that has the greatest potential for having people space out when he talks about it, I think, because nothing to do with you, but I think this, developmental bio, pulling back pulling back the cell membrane and peering inside, and seeing what's inside and trying to describe that to a lay audience and to make it so that they don't cloud over. I think that's very, very difficult. So with that as an introduction to make your job even more challenging, can you simply describe why it is that we need to know developmental bio and how a cell is really working in the inner workings in order to make progress here?
So there's no pressure on me at all?
No pressure at all. But I'm taking the pressure off because I'm telling you, anybody has tough time describing this. It's really hard to describe for a lay audience.
So I would take my starting point from something that Stefan said, that we sometimes forget that in the human body there are many parts of us that can regenerate or do regenerate most of the time. So Stefan mentioned that our skin is constantly turning over. If you've ever vacuumed under your bed and you wonder where all this dust comes from, well, unfortunately it's coming from you. Your skin is constantly shedding. Similarly, our digestive system. Since you've all probably had lunch, I can tell you that you'll lose 10 billion cells, that's billion with a B, cells from the lining of your gut every day, and they will be replaced every day until the day you die. And we take that for granted. And similarly, when you, for example, when you cut yourself, you injure yourself, your skin does something that it doesn't normally do. It starts to divide and repair and heal the wound, and we take that for granted too. Some of you may know that you can have up to 2/3 of your liver removed surgically, And in a healthy adult, that liver will grow back normally. So there are these processes of regeneration that are either happening normally or can happen sort of in a healthy adult after injury, and we sort of forget about them. And I think the job of us developmental biologists is to think about what is happening during repair. And you can really break that problem down into two simple processes. One is that if you want to repair something, you have to make more cells. The cells have to divide. And so understanding what tells a cell to start dividing and then to stop at the appropriate time is really important. And we steal, in fact, techniques that people working in other tissues, like the liver and the skin and the gut, we steal their techniques and apply them to the ear to understand division in the ear. Okay, so that's the first process. The second process is if you want to restore specialized cells, whether it is a motor neuron in patients with ALS, or whether it's a sensory hair cell in people with deafness who have lost their hair cells, you have to understand how to take one cell type and turn it into another. And again, by understanding the normal processes of cell turnover in, for example, our skin or our gut, or how our bodies respond to injury and make new differentiated cells that can carry out their mature functions. By looking at these processes and understanding how they can work in the ear, that gives us some insight as to how one day we may be able to repair the mammalian and the human inner ear. So that's really what a developmental biologist is trying to understand. How do cells divide? How do you make more cells? And how do you turn them into the right cell that you want?
That was an absolutely beautiful, do you understand what you just heard? What a beautiful answer that was? Write that down, whoever's transcribing. That was a perfect answer. Really, I'm going to claim it's mine and say it exactly that way.
I'm going to be watching CBS News later to see if you use
No, I had this idea. But that is making me think. I know it's not up your alley, but that's okay. There are genes, I guess, ATO1, and I guess now it's called MATH1, which is a gene that's been discovered that if it's there, suddenly, in and of itself, can make a hair cell appear in the cochlea, right?
Sometimes.
Kind of. Sometimes.
Yes.
Are you? What about that? That's gene therapy. That's another sexy term. So what about that? Can we just give people some of them genes that they need?
We would very much like to. So just to give a little background, as cells make decisions to turn into a particular cell type, they have to change the genetic program that is active in that cell. And they do it with particular switches. We call them transcription factors. The name isn't important. And what people have found is that one of the first switches that is turned on as a hair cell is made is a gene called ATO1. The name is not really important. But basically, this is a hair cell switch. Okay? And a variety of groups around the country and around the world have shown that if you artificially switch on this gene, ATO1, in inner ear tissue, it will cause that tissue to make hair cells. And indeed, those hair cells are functional. You can show by various methods that they are able to detect sound vibrations. So that's great. However, what seems to be the case is that as the animal ages, that gene therapy, if you like, becomes less and less successful. So you are firing the same gene into the same tissue, it's just a little bit older. And so the challenge, and I'll be talking about this later in the afternoon if any of you are still awake by then, but the challenge is to understand what is going on as the ear ages that makes it refractory to these sorts of interventions. So, science is often about, big triumphs and then huge disappointments, and we hope that the trend is gradually upwards. But in the case of ATO1 gene therapy, the initial results were very promising, and then we realized, well, hang on, it doesn't always work in older animals, but now the challenge is we need to figure out why.
All right, so thank you for that answer. I'm going to turn it over to Q&A, but before then, I'm going to just throw out there the 800-pound cochlea that's in this audience right now, which is, this is all beautiful, it's wonderful science, it's wonderfully interesting, but when? When are we going to have the application of, when does benchtop move to the bedside.
So I'm the 800-pound old gorilla. I'll give you my answer. A lot of individual labs have been working on this for 20, 25 years. We had to show that it happened and do a lot of work that we'll all talk about. What I think the answer to win is, do we bring more great scientists into this field? Is there the money to do it? Is the society or the public or wonderful people or wonderful donors willing to spend the amount of money to bring all, you just heard how my research relates to Stefan's research and how Andy's research relates to Stefan's research and vice versa. What we've formed here with the Hearing Restoration Project is a way to bring these all, if you will, under one hypothetical roof, to reify this collaboration and cooperation. If we can do this, okay, I think that within 10 years, we can find the pathway to restoring hearing in humans. That doesn't mean we'll have a drug at the bedside, but once we find that pathway, that mixture of genes and proteins that need to occur to make that happen, then Big Pharma will take it over. And they have very, very deep pockets, and it's a very, very big industry to try and cure hearing loss. And so I think that's going to depend on getting great scientists working together as teams.
And by the way, this is the new model that we're seeing with others, which is the collaboration between scientists, patients, pharma, business, academia, and the old model of eating each other's lunch. We're not going to allow that. In fact, you have to help each other make lunch for each other, and that's the way. And that's how you're going to get the money. And we see this with Stand Up to Cancer. We've seen this with Kathy Justy's and Multiple Myeloma Foundation, which has had an amazing success with this type of model. And I have to say, 50 million over 10 years, I'm thinking that is so little. That is such a little amount of money for something that's going to affect tens of millions of Americans. It's almost embarrassingly little amount of money. So certainly we need more support. Why don't we throw it out to questions from the audience? Yes, we have a mic for her. Hold on one second.
Hi, thank you. I'm Jonna Schachter. I'm the chair of the Hearing Access Program. And I have a question actually for you. One of the biggest issues is getting media attention. And I have written pieces on this, and one of what I keep being told is hearing loss is about them, not about us, and it's boring. And you can't see hearing loss. It's an invisible disability. Therefore, you can't show it on television. It's hard to show it. Their perception, it's an older adult issue when it's a teen issue, as you mentioned. How do we bring and make hearing loss sexy? And make it so that the media actually wants to do stories on it. I mean, we have induction loops all over the city, in every subway information booth as part of President Obama's stimulus package. Huge, 352. Not one state, there is going to be an article hopefully next weekend, but there has not been a single article in the media about that. And then that's part A. Part B, I was at the last NIH meeting with Dr. Rubel, and we talked about 10 years. And here we are 5 years later, talking 10 years. And the question is it always 10 years away? And I know that's a little tough of a question, but I figured I'd.
I'll take part A, you take part B. You want to do it the other way around?
Go for it.
Part A is a tough one, and it's frustrating for me. I mean, it's a 22-minute broadcast. So for the evening news, there are two types of stories. There's a story that's tied to a news peg. Something just came out. Tonight we're going to do a story of something that's just published today. Or it's an enterprise story. Like we did a story, a 6-minute piece on Alzheimer's, and it took us months to do. We went around the country, and those are much rarer. So in terms of the evening news, a national broadcast, it's very tough real estate, tough to get even. There are tons of stories that I want to get on all the time. And so it needs to be something where there's a news peg, something that was just published in the New England Journal that's seminal, and not just sort of basic science, but also a phase 3 trial that shows that something is actually working today. That's the bar. I'd love to you know, to be able to wax lyric, I find one of the great frustrations is a lot of things that I want to do. I want to talk about Haiti. I want to talk about Somalia. I want to talk about all sorts of things, and it's just tough to get that real estate there. So I mean, I share in that frustration. I would just say, keep trying. But
so I'll start off, and then maybe some of my colleagues will add in. I think your question is a really good one. I actually never said that we would solve it in 10 years at the NIH meetings. I recognized that this was a difficult problem. It was going to take time and money and new technologies, okay? But let's look at it. The war on cancer started in 1971 formally. Okay, for 30 years or so, people said, oh, we're not making progress. But people were understanding in their labs the cell cycle and so forth, research coming from yeast and from cells in culture and things like that. And all of a sudden, 10 to 15 years ago, wow, we started preventing cancers, and we started slowing them down such that very few people that are diagnosed early are dying from breast cancer or prostate cancer and things like that. So it takes time, and it takes a lot of muscle, okay? And what I mean by that is there are probably 2,000 laboratories in the United States alone working on cancer. There's 100 times the funding that goes into studying hearing, all aspects of hearing. So we have right now somewhere between 10 and 20 serious laboratories in the United States or in the world studying hair cell regeneration, okay? And a few $100,000, okay? So what we need to do is have a war on hair cells. Okay, and then it will move along. As a scientist, and as a scientist that actually had a role in starting this field, I'd like to see a cure in 10 years. I'd like to see a cure in 20 years. I'd also like to see a cure in 50 years or 100 years, because it's going to help people then too. Okay, what we want to do is move it from 100 years to being at the bedside in 20 years.
You know, it's also more frustrating in terms of the media coverage, or in terms of even people perceiving what's the advancing, how much is being advanced in the field. Cancer is not, there's no such thing as one type, there's no such thing as curing cancer. There are hundreds and hundreds of different types of cancers. And the mortality in men in 40 years has gone down from 22%, in women 14%. As Ed said, cancers are prevented, over 90% cure early. But this is chipping away at this cancer and that cancer, and little by little. So there are these successes that we report on all the time. We do stories on cancer all the time because we have this little success. It's tougher with hearing loss, isn't it? Because it's not as incremental. It's almost all your eggs are in this one vestibular and cochlear area. And once you crack it, you know, probably there'll be a tremendous breakthrough, but it's maybe not as incremental.
But it's going to be incremental. Go ahead.
But I think one thing to consider is also hearing loss is multifacetorial. It's not. It's in this way. It's similar to cancer. There are people in the audience very likely who are have hearing loss because of a drug overdose, or a gentamicin or aminoglycoside-induced hearing loss. There are people in the audience who very likely suffer from age-related hearing loss, and there are people in the audience who perhaps have a genetic disorder that leads to hearing loss. And each individual cause for hearing loss will have a different therapeutic approach. It's very I think it's unrealistic to think about that there will be the cure for hearing loss. I think there will be some cures for some forms of hearing loss that will enter the clinic earlier, and some hearing losses will be much harder to treat. And some of this, for myself, I had to realize this over the last 10 years, where if you would have asked me as a young assistant professor, how long does it take to cure hearing loss? And I bet I have said, you know, in 5 years we have a solution for certain things. It's totally unrealistic, and over time, I think I got a sense for the difficulty of the problem and for all the roadblocks and the issues we have to deal with. And I'm getting frustrated myself, how long it takes to overcome a single one of these roadblocks, and then you're over the hill and there's another one and there's another one. On the other hand, now we can see more through the fog. There's no roadblock hiding behind a bank of fog anymore. I think we can see a real path where we have to go and what we have to do. The problem is it's really difficult to assess whether it takes 10 years or 15 years or 20 years.
Nobody's going to ever be able to give you that. And that's
Right.
a good point. I'm glad. It's true that I said lumping them together, and it's true. There are many different types of hearing loss. We're talking about sensorineural here. There's conductive. There are things that maybe hearing aids work better for. But we're going to sort of the meat of the problem, where there's nerve damage. You were trying to say something, right?